Okay, welcome everybody to our June 17th IMAG MSM working group meeting. As usual, I need to remind you that the meeting is being live streamed on YouTube and is being recorded and will be available after the fact. As usual, Reinhard and myself are always available if there are any issues you'd like to address. Jim Sluka handles the website of things, and Bruce Shapiro you communicate with on a frequent basis. We're always available to talk. We have our usual communication channels. I'm sure we're all overloaded with communication at the moment, but I do encourage you to use and publicize our YouTube channel because we have a growing and valuable archive of talks that I think deserve to be promoted. So without further ado on that, are there any announcements, short announcements, or issues that people need to bring up?
I'd like to make a short announcement. This is Reinhard Lauenbacher. If you happen to be registered for the meeting of the Society for Math Biology that's going on now, today is the last day, and at the end of today, I'll be hosting a panel discussion on mathematics and medicine toward a roadmap with Susan Gregoric from the NIH, Mark Palmer from Medtronic, and Jane Bai from the Food and Drug Administration discussing the future. So I invite you to attend.
You want to put the link to that in the chat?
Well, it's a link that's only valid for conference participants. So I don't think I should be doing.
Can you put a link to the page, at least for the conference, that says what the event is about?
Absolutely, I can do that.
That might be helpful. Sounds fascinating. Okay, any other announcements before we move on? I appreciate the people who are sacrificing SMB time to be here. I know it's a tough job. I'd like to give our schedule for upcoming meetings. Next week, we have Lisbeth Garris, who's the head of the current incarnation of Virtual Physiological Human. Of course, that's a very major European undertaking in this area. And then Rufus Pollock talking about data portals and data management. We are going to have a change of schedule. On July 1st, we're going to have a longer meeting than usual, a 2-hour meeting. It's going to be open also to the other working groups. And this is going to be Grace Peng and Reid Chapman talking about the big NIH initiatives in artificial intelligence for part of it. Some people probably have been on the Bridge to AI workshops that have been going on this week with NIH. And we're also going to have a discussion specifically at the request of Grace on how to combine multi-scale modeling and artificial intelligence to deliver biomedical digital twins. And so I think that that's a topic of interest to many of us, and we're looking forward to that. We'll try to have more organizational details as time goes on. Grace requested that we do this soon, and so we're setting it up as soon as possible. That means July 15th is going to be when we're going to hear our journalist friends do the presentations that were originally scheduled for July 1st, and the original presentation scheduled for July 8th will be held as originally scheduled. We're still looking for suggestions for speakers. You could volunteer, make suggestions for July 22nd, July 29th, and Auburn in August. So please send us your ideas. As usual, rules of the meeting, please mute your mics during the presentations. Try to hold your questions for after the presentations. I'll give our speakers a 5-minute warning. Today, since we have our first speaker coming from Israel, where it's very late because as I was saying earlier, while COVID may have abolished distance, it hasn't abolished time zones. We're going to let them speak. First, we'll have a short discussion session after that, and then Chantal will speak and have the time after the regular 4 p.m. Ending time for the discussion for her talk. So we're going to have a slightly different format today from the usual one. And without more ado, I'll turn it over to Adi Stern, who's going to talk to us about vaccine breakthrough, which is, of course, a topic of great interest to many of us. And I will let you take control.
Great. I hope everyone hears me well. Let me just share my screen. Okay, this something strange just happened here. I hope this is going to be okay. Right, so here we go. And first of all, thanks to James and to Bruce for inviting me to talk. And my apologies to any of those who were last week. One of the costs, I guess, is of time zones and working from home is that one loses track of time completely. Right. So we're going to be talking today about increased breakthrough rates of SARS coronavirus variants in vaccinated individuals. Someone on the screen. And so let me just begin with a brief introduction, which I'm sure you're all familiar with. So just a brief introduction to the SARS coronavirus genome, which we all know was sequenced in January of last year, and immediately led to the realization that this is a coronavirus. The most similar strain of virus is from bat coronavirus, particular strain of a bat coronavirus, which is 96% identical, and so on and so forth. We see other strains of coronaviruses that are similar to what was subsequently named SARS coronavirus 2, with the addition of one common cold coronavirus, which is 50% identical, and there are an additional 4 strains of common cold coronavirus in humans, which are all related to this virus. And this is the genome of the virus, where I think the most important part of this genome is this region, or most important as in it generates the most interest, since this is the spike protein. And the spike protein is what generates the antibody response. It's also the protein that is responsible for viral entry. And so it really has been the subject of most studies on genomic studies on the virus, of this virus. And so we're going to be talking about mutations, and I just want to give a brief introduction about mutations in general. And so we hear a lot about mutants, variants, mutations in viruses and in coronavirus. And it's important to note that mutations are a natural part of the replication cycle of all viruses, because viruses have error-prone machinery. Every time they copy their genome, basically an error is introduced somewhere in the genome. And so in coronaviruses, in this coronavirus, we know that every second transmission, on average, a mutation is fixed somewhere in the viral genome. This means that if we look at the entire set of viruses that have infected the globe, there are probably millions, or maybe not millions, but many thousands of mutations that have occurred. And most mutations we observe are actually most likely meaningless. And when I mean meaningless in evolutionary terms, what we mean is that they're neutral. They have no effect on the function of the virus, on the phenotype of the virus. Evolutionary biology in general, most mutations that we observe are neutral mutations, and that this is actually true for almost any organism. And in fact, early on in the pandemic, this paper came out. We shouldn't worry when a virus mutates during disease outbreaks. And there was a lot of discussion trying to calm sort of calm the public down from this fear that occurs when we hear the word mutation or mutant. And so this was actually true for the beginning, or for most of the trajectory of this pandemic all the way up to somewhere around September or October of last year. And the problem is this word most, okay? So most mutations really are neutral or meaningless, but rarely they are meaningful. And so there is a small probability that a mutation will become meaningful. And when I mean meaningful, it means that it will, in evolutionary terms, it will, the virus that bears a mutation will have an advantage, a selective advantage, and will replicate better. And from our point of view, when a virus replicates better, that might mean either that it is transmitted more efficiently, or that it is able to create a higher viral load. And these two are sometimes related. And this also might mean that it might be more pathogenic. And so by meaningful, again, I keep, since I'm an evolutionary virologist, everything from my point of view is from the point of view of the virus. And of course, the point of view of the virus also impacts the disease in the human. And so skipping from mutations, I'll again just talk very, very briefly about what's called variants of concern. And so all these various organizations, WHO, CDC, ECDC, all define variants of concern, which is a combination of mutations that generates concern, as their name implies. WHO listed these 3 variants. Actually, recently a 4th variant was added onto this list, which I'm not going to be speaking of today, although it's generating a lot of interest. But so we have B1117, which was originally detected in United Kingdom, B1351, originally detected in South Africa, and P1 originally detected in Brazil. They've been renamed as Alpha, Beta, Gamma, and Delta. Delta is the 4th variant of concern, which was originally detected in India and is now also spreading both in the United Kingdom, in other countries in Europe, and also to some extent in the United States. But I'm going to be speaking mostly about these 2 variants for the reason is, for the very simple reason that these are the only variants of concern that were detected in Israel. And that's what we have evidence that I will show you about today. And so mutations in the variants of concern, the interesting thing, again, from an evolutionary perspective, is that these 3 variants of concern actually share quite a lot of different mutations. What we're seeing here is the spike protein, again, that very important protein in the virus that is responsible for viral entry into the cell, and that's also what our immune system or our antibodies detect. And we see that there are similar shared mutations in these variants of concerns. In particular, N501Y is one which was shared across all these 3. And another interesting mutation is E484K. And it's interesting because this mutation was actually identified early on, already about a year ago, before these variants emerged. This mutation was identified in lab studies as potentially allowing immune evasion. And so what's shared among all these mutations in the spike proteins is that they all increase ACE2 binding, and ACE2 is the receptor through which the virus enters, and they all reduce antibody binding, or some of them belong to category 1 and some of them belong to category 2. And that is what makes these variants of concern, because they all show signs of increased transmissibility, and there have been, as we will soon see, there has been some kind of conflicting evidence on their ability to evade the immune system. And so when these variants of concern came out, a lot of labs went on to perform something called neutralization assays. And so neutralization assays are assays where serum is obtained from people who were either sick with COVID or were vaccinated. And the effect of the serum is tested on the growth of the virus, or in particular the variant of concern virus, in tissue culture. And what you see here is something called plaque assay, where we, in these types of assay, we basically test how many viruses we see, how many viruses grew in the presence of the antibodies from the serum. And what these different assays showed is that there was some reduction in neutralization of B117, but it was a very mild reduction. There was a small reduction of neutralization of P1. And there was a medium to large 6-fold reduction for B1351. And so you see this drop in neutralization, which you see here of B1351. But actually, this very study, which was published by Pfizer, so one of the manufacturers of one of the prominent vaccines, shows that despite this drop in neutralization, there still is neutralization. So we see that everything is above the dashed line, and the dashed line represents some threshold that they defined of enough neutralization to do the job. And so there was a series of studies. There was a very large number of papers that all showed very, very similar evidence. And so when these variants of concern came out, this was sort of like a note of caution, but on the other hand, it was also positive news because it's not that neutralization dropped to 0. There was definitely some neutralization here. On the other hand, there have been some conflicting reports on the role of what's called T cells. And so T cells are another arm of immunity that is present in our body. And so neutralization assays are based on B cells or on antibodies, but T cells is another arm that is present. And there have been, throughout this year and a half of this pandemic, there have been several different reports that claim either that T cells don't play a role or that T cells do play a role in protection against coronavirus. And this paper actually showed, as suggested by its title, negligible impact of SARS coronavirus on CD4 plus and CD8 cell reactivity in COVID-exposed donors and vaccinees. And so what this paper actually claimed, and this is a preprint, is that the variants of concern should not be concerning at all, because the T cell response is completely maintained, and so everything should be fine. And so this is where we started our research, and sort of our question was, what happens in the real world? So we saw all these different lines of evidence, both about T cells and about antibodies, and they're all performed in the lab. But eventually these variants of concern are meeting entire human beings, and we wanted to understand, do these two variants of concern, again, which were the ones present in Israel when we started the study, do they evade vaccine protection in real world settings? And so this is the timeline of our study. Oops, trying to move this bar, right, here we go. And so this is, so our timeline was during the beginning of February till the middle of March, actually from the end of January till the middle of, beginning mid-March. And in this time, there were actually two important things were going on in Israel. First of all, this was the middle of the third wave of the pandemic in Israel, so case counts were very, very high. On the other hand, vaccination rollout had begun, and in fact, there were quite a large number of people that had been vaccinated, in particular vaccinated with the first dose of the Pfizer-BioNTech vaccine, because that's the only vaccine that is in use in Israel in this period of time. And so we realized that this is kind of a sweet spot for testing vaccine breakthrough, because there were high case counts then, but on the other hand, there are also a large number of people that were vaccinated. And so the way we designed the study is that we focused on purpose on the rare vaccinated individuals who became positive for corona. Okay, and so we focused on these vaccinated individuals who either received only one dose, so they were 2 weeks after the first dose, up to 1 week after the second dose, or were more than 1 week after the second dose. And we chose these criteria on purpose, because these were the criteria that were both used by Pfizer when they published their study, and they were also criteria that were used by the healthcare, the HMO that we worked with, which is called Clalit. It's the largest HMO in Israel, and so they actually performed a study that tested vaccine effectiveness in real-world settings using these very criteria, and so that's why we used them as well. So we focus on these individuals, and basically what we did is we performed a matched cohort study where every single vaccinee who became infected with corona was matched with a non-vaccinated individual who was also positive for corona, who was the same age, same sex, same address, same sampling date. And again, this is to create a control population and to ensure that we weren't seeing an outbreak that was in a particular city, and so we might pick up a whole lot of vaccinated individuals that became sick just because there's an outbreak of a given variant in the city where they live. And so this controls for it. And we performed full genome sequencing of the viruses in all of these samples, where we sequenced the entire genome of the virus. And these are our results. And actually the paper just came out a few days ago, so you're also welcome to look at it. And so first of all, we looked at the variant frequencies in Israel. And what you can see here is that the B117, the so-called British variant, literally took over the population of viruses in Israel. So when we started, it was at a frequency of 70%. And at the end of our sampling period, it looked like it was 100% in our sample, but our sample size was actually pretty small towards the end of our sampling period. But I can tell you that already that from further studies that we looked into, this variant really took over almost everything in Israel. And the second thing that you can see here is that what we call the wild type, which is actually everything that is not a variant of concern, it's a mix of different lineages, dropped. That's the gray line. And we can see that the B1351 variant, which was again originally detected in South Africa, was at a low frequency overall in the entire period of study, around 1%. And so this was reassuring actually, because we were happy to see that this variant, the B1351, which raised a lot of concern, originally was at this low frequency. But then when we broke this down into vaccinees and non-vaccinees, we already saw that something is going on here. And you can see that this larger orange proportion is present here in people who were what we call the dose 2 category, so those that were 1 week after the second dose of the vaccine. But this wasn't enough. We went on to perform a statistical test. We perform what's called a McNamara exact test here, where basically what we do is every cell here in this table is our matching. Okay, so we have 8 individuals here who, or 8 pairs of individuals here, where all the 8 vaccinees were sequenced, that we sequenced here, had the so-called South African variant, whereas all their controls did not have the South African variant. And so that's where our control scheme is kicking in here. And we see this ratio of 8:1. Okay, so 8 individuals who had the South African variant were vaccinated, as opposed to the opposite scenario, where we have only 1 individual here who had the B1 control, who had the B135 variant, whereas his or her matched pair did not have that variant. And so this is statistically significant, and this is the dose. This is the dose 2. So I have, the label is missing here. This is the dose. These are the dose 2 cases. These are the dose 1 cases where we didn't have any meaningful results. The sample was simply too small. And so we see increased breakthrough of the so-called South African variant after 2 doses. On the other hand, looking at the so-called British variant, B1117, we see that here we found a significantly increased breakthrough rates after the first dose. Okay, so we see this 26:10 ratio here. And again, this is statistically significant, which means that we see more breakthrough of the British variant after 1 dose. And this was interesting, and this result in particular I think was very interesting because it was very difficult to see from our original results because the frequency of the British variant was so high overall in Israel. When we broke this down week by week post-vaccination, we saw that there was a very particular pattern. So the breakthrough of the so-called South African variant was in this particular time frame, so 7 to 14 days post the second dose. We did not, or almost did not, see any breakthrough, or we didn't see any breakthrough 15 days and onwards after the second dose from the South African variant. And this was reassuring because it meant that what we think is going on is that there's a really very particular time frame where this where breakthrough is possible. And I think you might have heard from this a little bit from Ron Relo, if I'm not mistaken. So to summarize our findings, what we see here is we see reduced effectiveness after 1 dose, but the effectiveness dramatically goes up after 2 doses. We see vaccine effectiveness is lower within this particular time window of B1351. Nevertheless, this variant did not spread in Israel, which was very reassuring. And we asked the question why, and our interpretation is that either there's some evolutionary trade-off between vaccine evasion and ability to spread. So we think that maybe this variant is simply doesn't spread well. Or the second interpretation is that effectiveness is only slightly reduced, and that vaccination was able to halt the spread of this variant. And this was actually corroborated by a study published in Qatar, which showed that vaccine effectiveness is around 75% for this strain, which is still very, very high. It's low. It's less than 95%, which was originally reported, but it's still very, very high. And with that, I'll end and just thank all our collaborators on the study and the wonderful team who performed the study. And thank you for your attention. I'll be happy to take questions.
Please go ahead. Thank you very much for your patience with the snafu. Questions?
Hi, thank you for the very interesting talk. I had asked in the chat, so by same residence, I think you don't mean, or I don't know, do you mean the exact same address, like the control was in the same household? Or
No.
do you mean the same city?
The same, yeah, so we had a few levels. We had city and we had a larger sort of geographical region in Israel. We almost always managed to match by city, but in a few cases we weren't able to match by city. And so then we went to a slightly larger geographical region. But no, it's not the same. It's not the same household.
Yeah, that didn't seem to make sense. So
no.
Thanks for the clarification.
Yeah.
Other questions? I was wondering, because now you're seeing more reports of lower vaccine efficacy for some of the newer variants in the news. What the likelihood of do you have any way of estimating the infectivity of people who have vaccine breakthrough infections? We had a talk the other day about the very strong correlation between viral load and infectivity of individuals. And so I wondered if there's if you have enough cases that you could actually ask the question, are the people who are having vaccine breakthroughs less infectious than people who are unvaccinated but infected, or not?
Yeah, it's an excellent question. Our cohort isn't large enough to test this. So we had a total of 400 vaccinated individuals here. But what we did see, which was a little bit surprising, speaking of viral load, is that we saw higher viral loads in the B1351 individuals. But I'm reporting this very, very cautiously because, again, the sample size was small. It's not only is it small, but it's also biased because we know that the regulations in Israel were such that someone who's vaccinated doesn't have to quarantine after exposure. And so this means that people only went to be tested if they really, really felt ill. And so I think that biases our sample very much and might create a bias so that we see higher viral loads because of that. But that's all I can say. I know from other studies that yes, that it has been found that that vaccinees are less infectious and they transmit. There's a lot of different signs of evidence that I don't know in the context of variants of concern if this has been measured.
Were you able to pick up possibly also the issue of second infection after primary infection? Because there now also claims that the vaccines are more effective at preventing infection than having had the disease itself. And I remember fairly early on meeting two people who were both had COVID twice over a period of 6 months. Of course, that's an N of 2 among my acquaintance, but it interested me that if you were able to see anything related to people being reinfected after primary COVID infection.
No, so people infected after prime. I thought your question was different. They were actually excluded from our sample. So anyone who was previously infected shouldn't be in our sample. But no, I don't know anything else about that specifically. I will say that in general, I thought your question related to the transmission chains created by these infected people. And I was about to say that we don't know anything about the transmission chains created by any of these infected individuals because case counts were so high in Israel that all of the contact tracing actually broke down in Israel at this stage. So yeah.
Are there other questions? Well, I want to thank you again for all your patience and for staying up late. That was a fascinating talk. I look forward to seeing the follow-ups on that. It's going to be really, really interesting to see how these things develop.
I agree.
Thank you again. We appreciate you joining us.
Thank you.
Okay, so now we move to Chantal for our second talk. Are you ready to take over?
Yeah, I think I can, if I can share my screen. Oh, not yet. Let's see. Let me try and make this work. Okay, can you see the slides? Okay, so what I'm gonna try to do today is to a little bit describe how particles in the air, when they're inhaled, where they end up in the lung, because it seems like it's a expertise that not a lot of the people on the group, they are fantastic modelers. So maybe the combination will lead to nice things. But so the first thing I want to do is to actually, let's see if can I move my slide. Here we go. It's for those not familiar with the lung, give quick description of how the lung, the anatomy of the lung. And so once you breathe in the air that goes through your nose or your mouth and get to the back of your throat, it enters the main airway of the lung that's called the trachea, that's shown here on a cast of a lung of a human subject. And then it divides basically over a series of about 14 to 15 generations. The division is the dichotomous branching pattern. And you get first the two main bronchi that leads to the right and left lung, and then this process goes on. And these first 14, 15 generation of airways are called the conducting airways. They do not contain any alveoli. And the main role of these airways is basically plumbing to bring the air into the respiratory region of the lung, where you have your gas exchange, where the blood takes in the oxygen and remove the CO2. When you go further down the generation, you start reaching respiratory bronchiole, which is kind of shown on these figures on the right here, where you can start to see having a little bit of alveoli popping on these airways. And as you go still deeper in the lung periphery, then you start having what's called alveolar ducts that are fully lined with alveoli. There are about 300 million of these little alveoli in the lung. So this is a huge surface area. It's actually the biggest surface area of the body that's in contact with the outside world. If you would take all these alveoli and spread them on the surface, you would fill about half a tennis court. So it's a huge surface area for the respiratory region. But in terms of volumes too, the conducting airways is typically at the end of a normal expiration in an adult, about 150 milliliters, and most of the volume is contained in the respiratory region. And these lung morphometry is usually represented by what's called the Weibel's model. It's one of the most used models to do any modeling either of flow in the lungs or particle transport that's shown in the middle of this slide. In terms of gas transport, another important feature of the lung is that if you look at what's called the total cross-section as you go from the trachea deep to the lung, and that's referred to as different generations of airways, with 0 being the trachea, and you go all the way down and you look at the total cross-section, which is the sum of all the cross-section of airway belonging to a certain geometry, a certain generation, you end up with a very rapid increase in total cross-section. This is a log scale on the y-axis. And the consequence of that is that when you breathe in at a constant flow rate in the lung, the mean airway velocity in these airways drastically decrease from the first few generations going down to the lung periphery. So once you get into the respiratory zone of the lung, which is generation above about 14, you have very, very low gas velocities. And this is just because you have such a big cross-section area in this area, because you have so many airways in that given generation. Which means if you can calculate based on these morphometric data, how much time a particle that would enter an airway would spend in that specific airway. And because you have such a decrease in velocity, even if the airways in the lung periphery are a little shorter than the large airways, the increase in residence time is very important in the respiratory zone of the lung. And that's something to keep in mind when we'll talk about the gas, the transport of particles in the lung. This is an important feature of gas transport. So briefly describing the main mechanism of deposition when you breathe particle in the lung, there are 3 main mechanisms that are based on impaction, sedimentation, and diffusion. And depending on the particle size, these mechanisms will be more or less important and participate in the deposition process when you breathe or inhale these particles. For large particles, and when we talk about large particles in terms of particles that penetrate in the lung, it refers to particles that are larger than 5 microns. And these particles mainly will deposit in the lung through inertial impaction. And that happens when the particles cannot follow the airstream line because of their weight and size, and they will just keep on their trajectory and eventually impact on the airway surface. And that happens a lot at the back of the throat, because that's one of the biggest change in airflow directions when you breathe in, because you go from almost a 90-degree angle from the mouth, the air coming in, and then have to go down to the trachea. It also happens in the large airways at bifurcation when you have your flow splitting into the 2 daughter branches. The next parameters is gravitational sedimentation, and these affect particles mainly in the 1 to 8 micron particle size in the lung. It's due basically to the settling of the particles under the action of gravity. And this mechanism happen mainly in the medium-sized airway in the respiratory region of the lung. And finally, the third main mechanism is Vibranium diffusion. And this is for smaller particles, and it's just under the action of Brownian diffusion, these particles move around and eventually touch airway or alveolar surface and deposit there. So out of these different mechanisms I just described, impaction is a velocity-dependent mechanism because it's depending on the speed of the flow. And so these particles will mainly deposit in the first generation where mean airway velocity is the highest compared to the respiratory zone. On the other hand, sedimentation and diffusion are time-dependent mechanisms, meaning the longer time they can spend in an airway, the longer time they have to settle or diffuse and eventually deposit. And so sedimentation and diffusion are mechanisms that mainly affect deposition into the distal part of the lung. And so when you breathe in an aerosol, the place it's going to deposit in the lung, knowing the size distribution of that aerosol, it's an important parameter. And this shows you for very controlled experiment done in the laboratory, when you breathe in particle, and the range here, again, it's on a log scale from very tiny particle, the 10 nanometer here to 10 micrometers. What's the depositions? That means the amount of particle you breathe in of that size, how many are retained in the lung after a breath. And these, we can focus only on one of these curves. These are different breathing volume from 1 liter to 2 liter at a certain flow rate. And you can see that for large particles, you have very high retention rate or deposition rate. And this is due both to inertial impaction and to gravitational sedimentations. Then when you decrease the size, these mechanisms become less efficient and deposition decrease until you reach a minimum in that curve that correspond to the particle size that are not too small to really be affected by sedimentation or inertia, but are still big enough that diffusion is not really an efficient mechanism for this particle to deposit. When you keep decreasing this particle size, then diffusion takes over and you can have a lot of particle deposited. So nanoparticles tend to be that you breathe in, a lot of them will be deposited in the lung, and same thing for the large particles. So when you talk about aerosol in the air that you breathe in that could deposit, what really matters in terms of particle size that can reach the lung, or typically particle up to 10 to 20 microns, because anything else would be filtered by the nose. It will deposit there and not reach a deeper region in the lung. Another factor that you have to take into account is that deposition is also affected by lung disease. And this is because the lung mechanics and the size of the airways change with disease. Some can be obstructed, some can be lose elasticity or elastic recall in the deep region of the lung, like in emphysema. And this is just one example using particle with 1 micron that tend to deposit mainly by sedimentation in the deep part of the lung. And you can see here the deposition rate for normals, which is about 10–15%. And when you look at different group of subjects, from smoker with no symptoms to smokers that have small airway disease, asthmatic and COPD that both have constriction in the large airways and also emphysema in the lung periphery, you can see that you can almost double deposition or the retain of particles in the lung, meaning that compromised population with lung disease that breathes in viral particle around you will be at higher risk just because they can retain more particles and then potentially more viral particles. Another aspect to remember too when it comes to how many particles deposit in the lung, it's the local surface dose, because you also need some time to create an inflammatory response. You need a certain amount of particles at a specific region on the surface of the airways. And this graph is a little bit complicated to understand, but the main message here is that these three-dimensional graph shows you on this lung region here different depths in the lung, from region A being the large and medium airways. And as you go down to these regions, you go further and further into the lung, with the last region here really covering the alveolar region of the lung. The data are shown for three different particle sizes that are shown here on this axis. And this is basically the calculation of the local deposition dose per surface area. So remember, I mentioned at the beginning that the respiratory zone has a surface area of almost half a tennis court, while the conducting airways have a very tiny surface area. So even if you have less deposition in these airways per surface area, they still end up having a much higher local dose. And that's to keep in mind again, in terms of drug delivery, but also of inhalation of pollutants or viral particles. So how to link this to, so this is what's just showing the ratio of the surface dose from like the large medium-sized airways to a region in the respiratory or alveolar region of the lung. And you can see that for all particles, and especially for larger one, as a surface dose, you have much higher values in the large airways than you have deep in the lung because of that big surface area down there. So here what I'm trying to do is give a little bit of what we know about respiratory droplets, which has been a lot in the news. It was kind of nice that people actually recognize aerosol scientists for once. Now people know what I do. It's where these particles that you generate in the lung come from and how big are they. So they originate from several parts. They originate from the lung themselves. They also originate in the larynx area, but also in the oral and nasal passages. Particle size, these droplets that are generated are mainly water. Over 95% of the content of this composition of this particle is water. But the number of particles, the size, and the composition of these droplets are linked to where they are generated in the lung, what kind of mechanism generated these particles, and what kind of activity you were doing. If you're breathing, you're coughing, or you're singing, or you're just vocalizing, it will generate particles from different areas in the lung. There is also a very large variation between subjects in terms of the number of particles that's generated, even in healthy subjects. Again, it may explain why some people are super spreader and some are not. It may just be linked to how many particles or respiratory droplet these subjects are generated while they breathe or talk. And the particle size vary from small particles less than 15 micron, which is basically the particle size that could be re-inhaled and get deep in the lung of people around that particular subjects, all the way to particles that are much larger than 100 microns. Another thing to remember is that when you breathe out these particles, they come from a very super saturated environment with relative humidity very close to 100% throughout the lung. And once they are breathed out, they shrink rapidly to about on average a threefold value. And this depends, of course, on the temperature in the outdoors and the relative humidity on the environment you're in. But that's to keep in mind that even if you generate a particle that's larger than 15 microns and that would not penetrate in the lung, when breathed in, by the time they reach a person around you, it may have shrunk to 5 microns and actually reach and bypass the upper airway. So I tried to, and there is a very nice review that just came out, and I put the DOI down here on the slides. That's from where I took all the information to create this table. I tried to look at where the droplets are generated based on the different activities and what kind of mechanism create these droplets, and try to have an estimation of the size from what we know so far. So when you're just breathing, which is the first column, the origin of the droplets come from the small airways. And here the mechanism by which it happens is that when you get to low lung volume and you lose the elastic recall that keeps in the lung because you reduce the lung volume, and the elastic recall, one of the effect of it is that these small airways are kept open by the elastic recall or surrounding alveoli, keep them nice and open. When you get to a low lung volume, that mechanism is not there anymore and your small airways become more floppy. And when you age, it gets even more floppy, so that if you reach a low lung volume, the lining fluid on these small airways can actually occlude the airway and create kind of a bolus of liquid. So the following inspiration, what happens is that you reopen these airways and that bolus of fluid become a film that eventually bursts into aerosol droplets. They're first breathed in because that phenomenon happens during inspiration, but then on the next expiration, these little droplets are exhaled in the air. The size of particles that are generated from these small airways have been characterized to be between 0.1 and 2 microns. And it kind of follow a log normal distribution that's centered at about 0.7 micrometers. And in some studies, they try to estimate the volume of liquid that exhaled during just breathing, and at rest, an average value is about 15 picoliter per liter of air that you breathe out. That volume, of course, increases with exercise because you increase your breathing rate, and the amount potentially also create more turbulence because you increase your flow rate, generating more particles. When you cough or sneeze, the origin of the particles now comes mainly from the mouth, larynx, and large airways, and oral and nasal passages. And in terms of coughing, the mechanism of coughing, when you are about to cough, what happens is you first close your glottis, and your diaphragm, which is the main muscle of respiration at the bottom of your lung, contract, reducing the volume of your lungs. But because you're in a closed system, you increase the pressure in your lung. And when you cough, you have an expulsion of compressed air, which generates all kinds of turbulences in the airways, in the large and medium-sized airways, that can generate bursting of the lining fluid on these airways, again generating particles. And there is a very big range of particle size from less than 1 micron all the way to 100 micrometers. And it's the same phenomenon using during sneezing. Now, what happens when you speak or sing? Now, the site of generation of these particles is located at the vocal cord level and in the oral passages. And you create small fluid filaments in these vocal cords during singing or vocalizing that again burst into small droplets that you breathe out. You also have a fluid film in the mouth around the tongue and the teeth, and they will also burst when you speak. At the level of the vocal cords, it looks like the particle size is about 1 to 5 micron. And at the mouth, because you have that big film bursting close to the environment, again, the particle size seems to be pretty wide. Now, these respiratory droplets can potentially include variant, and that's what we are interested in case of transmission by aerosol during pandemics. And the viral load that you measure in these respiratory aerosol reflect the concentration of such particles at the level where the droplet were generated. It turns out that the AC2 receptor that were mentioned in the previous talk are mainly located in the upper airways and then deeper in the lung, which happens to be exactly the two main sites of particle generation, which is probably not good news in terms of viral transmission. There are some studies that have been published last year that showed that the viral load in saliva samples seems to be much higher than thought. The thought was that most of the viral load would be in the sputum, and these studies shows that you can have a lot of these viral particles in saliva sample up to from 10 to the 6 to up to 10 to the 9 copies per milliliter of fluid. The variant size report in the literature show a size for this of between 70 and 140 nanometers. So in theory, any particles that are larger than that size could potentially contain viral particles, those respiratory droplets. And the probability that those respiratory droplets would actually contain these variants is proportional to their volume. So 100 micron particles will probably get much more particle in it than 1 micron particles. But this has to be balanced by the fact that these large particles will settle once they are airborne much faster. And this is just an illustration I took from a position paper that was published earlier this year by the German Association for Aerosol Research that show you particle size from 0.1 micron or 100 nanometer, about the size of a variant, up to 100 micrometer, which is kind of the upper bound size of particles, respiratory droplets. It will settle in a few seconds, a 1 meter distance, while these small particles here can stay for hours in the air. And the best analogy to that is that if you're in a room at two opposite sides, and on one side you have a smoker, that's exhaled the cigarette smoke in the room, and the typical size of a cigarette smoke is about 0.5 micron, eventually you're gonna smell that cigarette smoke at the other end, because this particle will stay sustained in the air a lot. And so based on the flow in the room, this is gonna diffuse eventually, and you're gonna smell it at the other side. So the big particles would not be too much of a concern if you, hence also the concept of social distancing, so that most of the variants that would be contained in these larger particles will be basically below what you can breathe in and eventually contaminate people around you. But the 1 to 5 microns, those particles that actually deposit in the lung, can stay in for a substantial amount of time. And so those are more the particle of concern, in my opinion. So how can we, as this is more for modeling group, in terms of aerosol transport in the lung and deposition, there are several approaches that have been used over the years, and all methods have both advantage and disadvantages of everything you do. For the 1D models, which is the simplest model to use and typically solve a diffusion convective diffusive equation of concentration of aerosols through a model that described the lung, they're very easy to implement. They run very fast, and they can provide you deposition prediction for an entire breath over the whole lung, which is kind of nice. However, they typically lack regional information. And the model that are implemented out there that are available typically do not allow for varying the particle concentration or the flow rate over a breath cycle. You can just enter constant value for this prediction. A nice thing about these models, though, is that they exist not only for the human anatomy, but for a lot of animal models, like the mice, the rats usually use in preclinical animal models. And so you can use these models to try to understand or extrapolate deposition prediction from animal models used in preclinical studies to human application. The other type of models are based on computational fluid dynamics. They allow you typically to base your model, 3D model, on subject-specific information or anatomical features that make them very attractive, especially when you want to study disease or variability between subjects. And they also allow for very detailed deposition pattern within the air spaces. This comes at a cost, which is extensive computing resources to run these models. And because of that, they typically only focus on a specific region of the lung, which then is your model prediction becomes very dependent about the flow and the particle concentration that you impose at your inlet and outlet of your models that are not a closed system anymore. And an example of data that are obtained in these models, and this is a model of an upper airway, the mouth passages and trachea going down to the main airways that were performed by Ma and Luchin a few years ago now, but it's a nice demonstration of the information you get from these models, and especially the heterogeneity in deposition, which you can't get from 1D model. And you have the creation of hotspot typically at the back of the throat, and those are the large particles that deposit there by inertial impaction. And then also at the main bifurcation angle, you have a lot of particle depositing. We also, and I have performed a lot of these simulation in the respiratory zone of the lung, where we have implemented models that allow for expansion and contraction of the structure to mimic a breathing cycle that generate very complex flow stream in these models. And this is an example of 1 micron particles, which is a particle that has a high probability to reach that region of the lung. And again, you can appreciate from this simulation that there is a very heterogeneous distribution of deposition within these airways. And in a more complex model, you can even see that some airways may not even receive particles. So when you get estimate of deposition in the periphery of the lung, and it's an average over the whole lung, it doesn't give you a good insight on high spot that can actually result in deposition in certain area of the lung of a sufficient viral load to generate or start an inflammation process. And then the final aspect of modeling that's now more pursued by different groups is the multi-scale modeling, where you kind of combine both computational fluid dynamics and 1D model so that you can, again, predict deposition for the entire breath over the whole lung, but you can still include in your model some realistic subject-specific features that can affect the fate of inhaled aerosol in the lung. It still requires extensive computing resources, and also now you require much higher expertise in engineering to set up this type of model. And in these ones, the one that are the most pursued to date are models that link 3D models of the upper airway and the first couple generation of conducting airways. And the reason for that is this is the region of the lung that has a lot of turbulent flow potentially occurring, which will affect a lot the deposition patterns of these inhaled particles. And these 3D models are then coupled to either 0D model of airway and tissue mechanics to try to mimic lung disease, and or to what's called MPPD model that stand for multi-pass particle dosimetry models that also then can give you information of deposition in the distal lung. And I think that's all. What finished with this slide that just showed the multi-scale modeling approach, where you can start from CT images. And if you have more than one images, you can not only derive the segmentation and then come up with an airway model here into which you can do CFD simulation, but you can also, by image registration, get information on how during a breath cycle, your lung expand and contract, and typically in diseased lung, not in a symmetric fashion in all region of the lung, and input that information in your simulation to make them more realistic. And the last thing I wanted to mention too is, especially as a lot of the viral particle end up depositing in the upper airway, there is a large heterogeneity in deposition between subjects in this part of the airway. And one reason for that is the anatomy. And you can appreciate here that you have some airways that are larger than others compared to subjects, and different angle or curvature to go from the mouth all the way to the trachea. And these features will affect how many particles deposit there that could potentially affect how many viral particles end up in these patients, in these subjects. And I think I'm probably up for my 15 minutes. So that's all I wanted to say, but I just want to acknowledge this is not just my work. It takes an army to do this work over the years. And I particularly want to acknowledge the current collaborators, Wanjun and Azadeh in my lab, and the Pacific Northwest National Lab in Washington State, and Applied Research Associate Bauman and Owen, which participate in this current funding today with the IMAC. And so I'll stop here.
Okay. Thank you very much. We're a little bit behind, but I think it's fine. Time for some questions and discussions. That was very illuminating to me. I really appreciate your going over the basics very much. I can certainly ask a question if nobody else wants to get started.
Chantal, can you talk a little bit about the complexity of the calculations?
For the complexity in the multi-scale modeling?
Yes.
So there we typically use, and now what we're trying to develop, it's using the OpenFOAM platform. I don't know if you're familiar with that platform, but it's a freeware that allows you to compute flow field in these models, and it's basically based on solving the Navier-Stokes equations that describe the flow transport. And then you have the complexity in the upper airway comes to choose the right turbulent models that will actually reflect the flow the flow distribution in the upper airway. And then you typically couple that. You have two ways you can do it. Either you use a oh gosh, I'm blanking on the name now. You link the aerosol transport within the fluid and you solve for everything at once, or you first solve for the flow and then you use that information to use a deterministic equation to simulate the particle transport. The complexity or the time, the resource needed comes mainly from how complicated or how complex you want to describe both the aerosol and the flow pattern. If you do a constant flow, then it's a steady state and it takes much less time. But it turns out that actually the shape and of the flow form of your breath cycle actually do affect particle transport. And so you become in an unsteady simulation, and that's when time goes up. If you wanted to do, so there are about, at the generation of the end of the conducting airways, you have about 50,000 airways that leads to 50,000 different units of respiratory. So you can't compute the whole thing. So you have to make compromise. And then you linked either convective diffusive equation that to describe in a one-dimensional fashion the transport and deposition then in the deeper part of the lung. When I did the simulation in the lower region of the lung, and then you change also the you expand and contract your model during a simulation, then you again increase by a factor, an order of magnitude, your computation time. So it depends what your question is and what detail you want. You can't have it all. So it would take unrealistic computing resources at this stage. I don't know if that answers your question.
It does, thank you. I might follow up with you. We have some specific questions.
Okay, sure. I'm gonna move this here because I feel like I look away from you. I'm looking at the pictures of people.
Ruchira, you have a question?
Yes, thank you. That was quite interesting and illuminating. So one of the papers you linked by Stadnitsky,
yeah,
at least in the abstract, it seemed to conclude it was comparing breathing, coughing, sneezing, and speaking, and it seemed that it was targeting speaking as being most likely, according to these models, to produce the particles in the sizes of interest. So one question is whether you are agreeing with that or whether you assess that that is a correct assessment. And the other question about that is that it seemed to be putting speaking and singing in the same category, whereas just now you were talking, and earlier you were talking about the breath cycle, and the breath cycle is different in singing. We're taught to, when we sing, to breathe in, stomach out, and do diaphragmatic breathing and so forth. So how would that affect this fluid dynamics of these aerosols? So
to answer the first question, yes, I think singing and speaking are the activity most responsible for transmission. And the reason I'm saying that, it's because the particle that's generated comes from the upper airway and the vocal cords in a size that can be breathed in back and actually penetrate in the lungs or in past the nasal passages of other people. Because like at the vocal cord, if you remember from that table, the particle size generated is about 1 to 5 microns, which is a perfect size, 5 micron to impact in the throat and the 1 micron to be breathed in the respiratory region and deposit there, which is the 2 region of the lung where the AC2 receptors are located. It seems there are none of such receptors in like the conducting airways, like the plumbing between the larynx and the upper airway and then the respiratory region. So yes, I do think that's the one main reason that speaking and singing are the most responsible. And it completely fits with these first outbreaks at choir practices and things like that. Your second question was, oh yes, what was the position?
That singing is different than speaking.
Yes, oh yes,
the breath
yes.
cycle is different.
Correct. However, because the particle of interest that could get viral load comes from the upper airway, it doesn't really matters. The generation of particles that are generated in the small airways here are not important. It's the size that's important, and it's not from the person that speak or sing, it's for the person around you that breathes in the respiratory droplet you just generated. And 1 to 5 microns seems to be that perfect size. I mean, it is that perfect size. It's actually the typical size of any aerosolized medicine that people use for asthma or any other pathology. The typical size distribution of a drug is 1 to 5 micron because it's the one that actually penetrates in the lung. The good news is that if you find a medication to treat COVID-19 and you administer it by aerosol generation, it's actually going to deposit where the viral particle deposited in the first place. So that may be the only good news in that whole story.
Thank you.
I had a question, which is, if you think about spread of the virus within an individual, you could imagine the virus moving in the tissue. You could imagine the virus moving in the mucus layer. But you could also imagine re-aerosolization and spread of the virus within the lung. And I guess if you do steady flow calculation, you're not going to see that. But in a breath cycle calculation, what's the likelihood, and do you know anything about how the pattern of redistribution? Because there you don't have the shrinking of the particles. Even very small particles will stay in the alveolar region.
Yeah, that's a very good question. Actually, I've been talking with Greg Forrest on this, trying to get some modeling to answer that exact question. And I think what we believe, and if you remember when he presented a little bit of his work now, maybe a couple months ago, and he tried to look at dispersion of viral particles on cell just by being propagated through the lining fluid was not the answer, that if you have something deposit in the upper respiratory, it would not reach the deep lung. So there must be other mechanism. And so that's one thing that you could imagine. It's that if you talk or you generate this particle even in the upper airway, when you breathe, the immediate breath that follows that, when you breathe in, you still have a little bit of these particles in your nasal cavity that you just generated, and now they are the beginning of your inspiration, so they're pushed all the way deep in the lung. And you can do that. Typically, an adult breathes in and out about 10,000 liters of air per day. So even if you only get the last 3 milliliters of that expiration back in your lung, which is probably, I don't know, a very tiny portion, but even if you only have 2% of 10,000 liters that you breathe that can reach back in the lung, that may be you're completely correct. That may be a mechanism by which you can potentially infect yourself even more by moving through that respiratory droplet generation and rebreathing them right away deep in your own lung. Yeah.
I guess, but you think that comes from mainly upper airways, because
well, no, I think
what's the correct term? Is it expulsion fraction? What's the correct term? You don't eliminate all. You don't expel all the air in your lungs when you exhale.
So when you breathe now, normally we call that a tidal breath. So tidal breathing, it's like about 400 to 500 ml every breath. Now at rest, everybody here, that's about what they're breathing. But you're right. The thing in the small airways is that for the 1 microns, you need a lot of them, because every droplet would have less viral particles. And I'm not sure we know what's the threshold you need to actually generate this replication and generate an infection. But you're right that even during breathing, especially in compromised individuals that have a loss of elastic properties of their lung and have much more floppy airways, they can have that film bursting happening almost at each breath. And definitely severe COPD patients have that. And then if you have even a little bit of these particles, you could potentially imagine that at each deep breath you take in, you get these respiratory droplets you just generated deeper in the lung, and a fraction of them will be retained because they'll deposit.
So are individuals with defects like cystic fibrosis, is it with defects in the mucus that could be cause additional problems?
Yes, and there it seems like, because then you change the viscosity of your lung fluid. So I'm not quite sure what the droplet size and generation is, because now it's much harder, even with shear stress, to get droplets generated from a lining fluid because it's much more viscous. But that's a good question. It seems like they were not especially more affected by the COVID from what I have read, but I'm not really up to date on the topic. It may also well be that because they have so much issue with their lungs, they have been much more cautious about what they were doing. So I'm not sure it's because of the viscosity properties of the lung fluid in their lung, or it's just because they consciously didn't try minimize exposure.
Presumably a ventilator also gives you an unnatural flow pattern.
Correct.
And changes the viscosity of the fluid as well.
Yeah, because there the problem with ventilation, it's the drying of your airways. That's very true, yes.
So I have a question. At least once, I think twice, you made reference to individual differences.
Yeah.
That there's huge I don't remember if you said huge or major or significant. And so if we look across a bunch of people and you want to say, stay away from James, don't go anywhere near James. He breathes different. James is a what breather compared to me? Do we have a language and do we have measures in respiratory medicine that could even begin to classify people? And then you pile on top of that, well, it depends whether I just ran up the stairs or whether I just woke up. So I'm hugely different from James, the way I breathe. And James and I, maybe when I run, I look like James when he sleeps.
Mm-hmm.
Do we even have a clue?
Very little.
Of enough useful information to talk about how you predict transfer of a virus?
Very little. And the few studies that are there, and I think some actually of the reference are in that paper I was referencing to earlier, there can be an order of magnitude difference in the number of particles you produce between two subjects that are healthy. So people that are sick generate more particles, which is bad news in terms of transmission than a healthy person. So there is a huge variability there. There is not that many studies out. The other thing too, it's a lot of the studies that try to also characterize the size distribution of those particles that were generated tends to say, and you see that in a few papers that came up since COVID, is that they assume a distribution that starts at 0.5 micron. And the reason for that is that the experimental data in the literature were using devices that were not able to measure below that threshold. But that doesn't mean you have, we've started here a preliminary study at UCSD in collaboration with some colleagues in the Department of Chemistry that have sizers that you can go down to nanoparticles. And we have so far we only have 3 subjects, which are the investigator, because during COVID-19, no human research group will ask us if we can generate aerosol in the air in a room. They won't give you authorization to do this RB. So it's only on us, the co-investigators. But even between 3 individuals, we have largely different number of particles generated. And we do it just during breathing. And we read all the same text that's used in linguistic. We collaborate with a linguist. There is a whole piece of literature there looking at how you move your vocal cord, or what sound generate what kind of volume, something I was completely not aware of 5 months ago, but now I've digging into, I came up to that literature of which sound produce the most particles.
So if I want to have a digital twin of me that was capable of assessing my risk of a virus or would all those things have to be known about me in the twin? I mean, if I don't know what those things are, then...
Yeah, well, I guess, I think in terms of viral load, you need to know what kind of amount of particle you produce, because that's completely going to determine how much viral load or viral particle you can potentially make airborne in your environment.
Now that's different. That's that's my ability to transmit it more than my ability to get it. True?
So to get it, it's how much, well, no, if you generate your twin, once you have a particle size and you breathe it in, then it's always, it's gonna be, it's physics, right? Then
we would have to know for me,
yeah,
how I differed from every other human.
Yeah. And my guess is the biggest factors that are important is the anatomy of your upper airway, where a lot of particles are trapped, and your ventilation capability, which is if you have any lung pathology, that's something you would have to put in a digital twin type of model, or the healthy lung versus an asthmatic wouldn't have the same lung mechanics and somebody that has COPD or CF patient. So that's the kind of information you would have to put in.
I used to sing with an opera chorus. And when I think about that breathing and the difference, whoo!
Yeah.
Yeah. All right. Fascinating. That all of what you did is part of how it's like, if I have to use albuterol, presumably the particle sizes are designed to be trapped in the upper respiratory system. When I use Advair, those particles are designed to go down deeper into the alveola, and that's how that science works. Fascinating.
Yeah, there are people that make their career out of designing a new aerosol device so that it match the application. So you have all kind of aerosol device you can use, from meter dose inhaler to nebulizers, and they all kind of get.
What's fascinating is your topic today, and about a week ago, I started wondering about my Advair and how it really worked. and I learned a lot today. Thank you.
Okay, we're coming to the end of our time. If there's one quick final question, if not, we should thank Chantal for a very stimulating talk. And I appreciate. I think, Adi, it's interesting in the sense that I wish there'd been a third talk to connect the infectivity with the right. There was one missing piece to connect the two talks. But both very stimulating. So
I'd
thank
also
you.
I'm sorry.
Go ahead, please.
I'd like to suggest that if you have friends that are part of our group or not, that Lisbeth's presentation next week could be really interesting and have lots of opportunities to start building connections with the European efforts in silico medicine. So I would request that you send a note to people you like who might be interested and ask them to join that meeting.